class. जी बेटा, आप देख रहे हैं स्क्रीन इज देयर.
Yes, sir.
Okay, fine. So today we will talk about our new topic, that is nucleus, because in general sense we have studied about it, that whether it is a nuclear double membrane or single membrane or without membrane. So now we will discuss few organelles in upcoming topics, where we will try to understand what is their structure and how uh, they are performing their function in the cell. So, for
Okay.
example, the very vital organelle, sorry, very vital sub cellular structure, that is nucleus.
Yes, sir.
That is nucleus. Nucleus. So, it is one of the largest organelles. We can say it is the largest one, the most part of the cell, not most, but at the we can say as compared to other organelles, it has occupied uh, more space, right?
Yes, sir.
So we can say it is the largest organelle,
Yes, sir.
and definitely it is present in eukaryotes. We are not talking about the prokaryotes right now. We are discussing, because I have just named it nucleus. It means it is present in an envelope. So we can say it is covered with the nuclear envelope. Another point that we can add here, that point is covered with
A nuclear envelope.
nuclear envelope. And what was that envelope? I have told you yesterday there were two membranes which actually wrapped around the material which is present in the nucleus.
Yes, sir.
So, like, uh, for example, like this double membrane structure.
Yes, sir.
So, what it is, what it contains, definitely it contains genetic material.
Yes, sir.
Genetic material. In what form? That is in the form of chromatin. We use the word chromatin.
Noted.
Chromatin. Chromatin.
Yes, sir.
So, what is chromatin actually? Chromatin is actually, it is the combination of two things. One is DNA and the other one is protein. We will talk about this histone proteins. There are specific proteins which are present here. By
Yes.
the combination of this chromatin is formed because the DNA thread that is wrapped on the proteins, on these histone proteins, and form a very condensed structure that is called chromatin.
Yes, sir.
So, and both of these structures, they form a very regular structure that we mostly observe in the cell, that is chromosome.
Yes, sir.
Chromosome. So this coloring body or this chromosome is formed by the combination of DNA plus protein. So it is also present. And now
Yes.
the question is, what is its function? Definitely it has, if it has a chromosome in it, so its function is, because it is the brain of cell. We can
Yes.
say it is the brain of cell, means it has the control of all the activities which are taking place in the cell. For
Yes.
example, it control, we can say, control cell activities.
Okay, sir.
Control cell
Selectivity.
activity. Like what type of activities which are taking place in the cell? So there are many. For example, the very basic and the very important, that is metabolic process. Metabolism. It means the cell has to perform some activities, like it has to break the larger molecules into smaller ones, and it has to synthesize by using smaller molecules to form a larger ones. So we know the it is basic metabolism is actually what it is, the combination of all types of chemical reactions which are taking place in the cell. That is what we call metabolism. So it is of two types. We know one is anabolism and the other one is
Anabolism.
catabolism and catabolism.
Yes, catabolism.
Right? So catabolism, what we are doing in case of catabolism, we actually divide the larger molecules into smaller ones.
Yes, sir.
Right? So in
Yes.
case of anabolism, what we are doing, we are
Combination.
actually, yes, we are actually combining the smaller molecules to form a larger ones. For example, in you have a starch. If you are breaking into the glucose, it means you are catabolizing,
Yes, sir.
right? So if you are combining the sugar molecules like uh, glucose and to form a larger ones like starch or glycogen, so you are actually doing the anabolism. So this is
Yes.
how all these and many other functions. This is one of the functions that we have discussed. Respiration is going on there.
Yes.
All the information, you can say how that all is going on, because that's another process we will study about it in detail, like transcription and translation. This is a central dogma. We use the word central dogma for it. For example, if cell, there is a mechanism how this nucleus communicate with the cell. So it communicates actually by making or by synthesizing a messenger RNA that provides message, that has message, and that provides that message to the required, for example, ribosomes. That is
Yes.
providing that ribosomes, and
Yes.
ribosomes are the, yes, they are the factory to synthesize protein. So they will
Yes.
synthesize a specific protein which is required in the cell. So this is how it performs its function. Uh, Another thing, that is 2.5 micrometer size nucleolus is present inside the nucleus. 2.5. We can just, I just want to show you that uh, there is a specific spot. I have told you that in case of nucleolus, there is no membrane outside. If this is a nucleus, so for example, this is a nucleus, so there is just a spot inside, dark spot like this, right? So
Yes, sir.
this dark spot is actually its total size is 2.5 micrometer. Are you getting?
Yes, sir.
So it is present in the nucleus. So it is without membrane organelle. Remember, there is no membrane outside it. I have told you yesterday. What is the composition of this nucleolus? Now the question is, what is its composition? Uh, Let me give you another slide. Okay. Uh, Now we're talking about that. What is the composition of this nucleolus? Composition of nucleolus. Free space issue. It's okay. Acha ji, what is its composition? Uh, Ribosomal RNA, right?
Yes, sir.
Plus protein. Remember, right? So
Yes.
ribosomal RNA, that is uh, ribonucleic. We can say it's definitely it's ribonucleic acid. So that has also protein with it. So what is the function of it? Function of nucleolus. Function. Function of nucleolus. So what is its function? Its function is to produce ribosomes. Produce ribosomes.
Sounds.
Got it? So
Yes.
these ribosomes which are synthesized inside of this part of the nucleus, that is nucleolus, and from where these ribosomes will migrate, or they will transfer to the cytoplasm, and they will pass through a specific pores which are present at the nucleus. We always say nucleopores are specific pores. I have told you yesterday, two membranes, they converge on the specific point.
Yes,
There
sir.
they form a pore, and that pore is actually used to uh, for the, you can say, for cargo, for something to getting in and something is getting out. So that is the medium through which the materials is moving on the both sides.
Yes.
So this is about nucleus. So बस ये हमारे पास कुछ बेसिक इन्फॉर्मेशन इसकी होनी चाहिए. नाउ लेट्स डायरेक्टली हम मूव करते हैं जी राइबोसोम्स की, बिकॉज हमने राइबोसोम्स को डिस्कस किया है. सो लेट्स टॉक अबाउट द राइबोसोम्स.
Okay, sir.
राइबोसोम्स, हमने इसके बारे में एक चीज पढ़ी थी yesterday. What you have studied about ribosomes, what type of, where we categorize this ribosome, whether it is a membrane bound structure, single membrane bound, double membrane bound, it has no membrane. So
No,
where we categorized yesterday, what do you think?
no membrane.
Yes, without any membrane. So ribosome has no membrane. These are very tiny structures, granules sort of structures which are present in the cell.
Yes.
So first thing that we can write here about ribosomes, that is without membrane.
Non-membrane bound.
Out membrane. Got it. So uh, where they are synthesized, we can also add another point here.
In the
Where they
nucleus.
are synthesized, the nucleus. Nucleus. Okay, so and how they will move from the nucleolus to the cytoplasm? Through nuclear pores.
Yes, sir.
Nuclear pores. That's another point that you can add. So uh, ribosomes in eukaryotic cell. So definitely ribosomes types are actually changed in prokaryotes and eukaryotes, and this is a very important factor by which we can differentiate between prokaryotes and eukaryotes. Another concept that you are going to understand here. Ribosomes in case of eukaryotic cell. Let's talk about the ribosome which are present in the eukaryotic cell. So uh, ribosome, ribosomes. Do we have an idea what type of difference it will be here in both of these prokaryotes and eukaryotes?
Yes, sir.
What type? Uh, what would be the difference? If you
Size.
have your O levels, so there you must study about it, that how they differentiate from each other.
Size.
So the one thing is, in case of eukaryotic cell,
70s.
yes, 70s, and that's another difference. We will talk about that. But before that, we should know one thing. That is, in case of eukaryotic cell, ribosomes can attach to the endoplasmic reticulum. Yesterday, I think I have differentiated between rough endoplasmic reticulum
Yes, sir.
and smooth endoplasmic reticulum. Why they called rough and smooth? Because of the presence of ribosomes. If the ribosomes are present on the endoplasmic reticulum, that is what we call rough endoplasmic reticulum. We will have structures like, for example, if this is a nucleus and you're observing some outside endoplasmic reticulum structures, like these are the structures which are present outside. So on these structures, you will have small granules, number of small granules which are spreading on the whole structure. So like this, it will form a structure that is what we call rough endoplasmic reticulum. Got
Yes.
it? So, and those structures like this, like this, like this, and these structures do not have any type of small granules or ribosomes. So if they don't have any ribosomes on it, so we call them, what do we call them? Smooth
And
endoplasmic reticulum. So this is the difference between these two types of reticulum. And this is the one way the ribosomes can present. But another way is they may also present freely in the cytoplasm. Remember, this is the second point that I'm adding here. That is, uh, they are freely, freely present in, freely present in endo, oh, sorry, in cytoplasm. In cytoplasm. Yes, in cytoplasm. So it means the ribosomes, which were synthesized in the nucleolus, they transferred from the nucleolus outside of the outside to the cytoplasm by the nuclear pores, and now they are present in cytoplasm. They have two positions. One is they are present on the endoplasmic reticulum, and that reticulum, and to form or to make it rough endoplasmic reticulum, and the other way of these ribosomes to reside in the cytoplasm, that is to present freely, and they are floating in the cytoplasm, right? But their
yes.
function is very same. Function is not going to change because of their position. Function is only the function to synthesize protein by getting message from the nucleus.
Yes, sir.
You know, uh, just like a card reader or something, like
Yes,
you
sir.
can read, they are just acting here as a reader. Message will come from the nucleus, and they will just read that message and to trans and give that message to the next machinery, next part of the factory, where transfer RNA is there, and they will get specific amino acids, and they will bind to each other. And so this is how the protein is synthesized. We will study about this protein as well. So now we are coming towards a very short difference between eukaryotic cell and prokaryotic cell. Eukaryotic cell and prokaryotic cell. Eukaryotic cell and prokaryotic cell. Trying to give it should be on your side instead of raising from here. Anyway, so difference types of cells. We can just divide here the types of cells.
Yes, sir.
Types cells. Types of cells. Uh, let's suppose definitely we always divide these all types of cells into two types. One is eukaryotic, and the other one is
Prokaryote.
prokaryotic. Prokaryotic. Got it? So first difference. First difference is we will start from the nucleus. There is true nucleus present in the eukaryotes. What do
Yes,
you mean by this true nucleus? We have studied about the whole structure of the nucleus.
yes, sir.
There would be a specific double membrane structure there. There would be a nucleolus in it.
Yes, sir.
There would be DNA present inside of the nucleus. So this is the characteristics and the features of the true nucleus. Are you
Yes,
getting?
yes, sir.
While in other case, in case of prokaryotes, no true nucleus. No true nucleus. Got it? So
Yeah.
that's the main difference. Now comes we will talk about the because in case of eukaryotes, what is the main uh, you can say classes are the types in eukaryotes. Definitely, there are two types of cells which are present in the eukaryotic cell. One is animal cell. Uh, animal. Animal cell.
Yes, sir.
The second type of the eukaryotic cell is plant cells.
Yes, sir.
So these are two types of eukaryotic cells we commonly discuss. And as you just told a few minutes before that the ribosome which is present here, that is ATS.
Yes, sir.
Because will the subunits which are actually by which the whole ribosome is formed, that gives us uh, ATS. S is actually Spiritberg. It's who's a scientist because he used actually and named these two types of subunits. It is the sedimentation unit. You know, so what is sediment? Something that is taking time to sit on the surface.
Okay,
That
sir.
is what we call sedimentation.
Okay, sir.
Sedimentation means like uh, we commonly use this term in geology and somewhere like if the particles which are flowing with water, if they are getting down on the
Yes.
surface and they are forming a layer, that is actually sedimentation, sediment. And uh, this is the speed of sedimentation, which unit is getting first on the surface,
Okay,
that
sir.
is we called. And the scientists who use this method to define or to differentiate these subunits, his name was Swedberg. So we use his name and giving it unit S. So on the other side, DNA present in the cytoplasm. There is no true nucleus. It means there is no true nucleus. So where is DNA? DNA, definitely DNA or RNA. This is the very basic part of every cell. Every cell will have this, but it depends. It is eukaryotic or prokaryotic. Definitely in eukaryotic, the genetic material or DNA will be present inside the nucleus. While in case of prokaryotes, where there is no true nucleus, so this DNA will be present in the cytoplasm. So we can write another difference here, like what is that? DNA present in DNA present in what? In cytoplasm.
Class.
Got it? So
Ye-
where is the DNA present in case of eukaryote? That is present in the, the nucleus.
Nucleus.
Are
Yes,
you
sir.
getting?
Yes, sir.
While in case of prokaryote, DNA is present in the cytoplasm. That's
Cytoplasm.
the difference. And what is the size here? Size of this ribosome is
70
70s.
yes.
These are few differences that we are learning here, these two types of cells. One is eukaryotic through nucleus, and the main categories of these eukaryotic cells are animal cells and plant cells. And the ribosome difference here is ATS in case of prokaryotes. So I have studied about. So
Yes.
types of ribosomes. It means we can differentiate or we can categorize the types of ribosomes. Here we have just studied the different types of cells because these ribosomes are actually also differentiating these two cells. One is ATS and another is 70S.
Yes.
So we will further study about the types of ribosomes or what is the main difference uh, in these ribosomes. Okay, so types of ribosomes. So ribosomes. Yes, there are two types of ribosomes. As I have told you, on the basis of size of uh, unit, particle size based on its sedimentation rate. I have told you
Yes, sir.
particle size. How will you differentiate? Because of the on the base of the particle size. In
Okay, sir.
particle size, how we will determine this particle size? Based on sedimentation
Yes.
sedimentation rate. Sedimentation rate means this is a surface
Yes.
and these are one particle and this is another particle. How much time they're taking to get on the surface? How much
Okay.
time they're taking to get on the surface? This is this is sedimentation to
Okay,
sit
sir.
on that surface. Got it? So ribosomes, one is, as I have told you, one is 70s and another one is
It is.
8s. So uh, it is about, the 70s is about 18 to 20 nanometer.
Okay, sir.
80s is about up to 25 nanometer. This is the one.
Okay, sir.
Okay. So nanometer, it is definitely, one is smaller in size, another one is larger in size. So uh, now ribosome, as I've told you, ribosome is composed of two main components. Ribosomes of two components. One is R RNA plus protein,
Yes, sir.
right? This is
Yes.
what we call ribosomal RNA, because this is a specific RNA which is present in the ribosome, and this is the one, like we will study about it, like how they are present there. Uh, there will be a layer of RNA on which these two subunits are combined with each other. So this is how they are together. One unit is, one of them is larger and one other one is smaller. So function is, what is the function of ribosomes? Its function is
Protein
to
synthesis.
synthesize
Protein.
what? Proteins. Its main function is to synthesize protein. So uh, okay. Now, in mitochondria of the animal cell contains 70s ribosomes. Chloroplast also contains 70s ribosomes. And mitochondria and chloroplast both have their own DNA. We have studied about it, but now we will start another organelle. Anyway, this 70s is present. Remember, this 70s is present normally where?
Uh,
Normally it is present in
prokaryotic cell.
in prokaryotic cell. In
Prokaryotic cells.
prokaryotic cell. Now, anybody asked you this question, where this 70s is also present? Like in case, one case is defined case, that is the prokaryotic cell.
So
But
chloroplast
is there, yes,
and
other two,
mitochondria
two mitochondria
and
and,
chloroplast.
so these are the two structures in which this 70s ribosome is present. As
Yes.
we know, both are self-replicating or self uh, uh, protein synthesizing
Yes,
organelles,
sir.
and so that's why this is present here. Because it, what does it mean if the ribosome is present somewhere? What does it mean? It means that has capability,
Nucleus.
the ability to synthesize protein. It means that
No.
mitochondria and chloroplast, these two organelles are subcellular structures.
Yes, sir.
uh, 10 minutes are remaining. Okay, fine.
Yes, sir.
It's okay. So I think we can also study one more topic, that is mitochondria. Mitochondria is also very simple. We just need a very short introduction of this topic, that is mitochondria. Mito What is it? It is a powerhouse of the cell. All the energy, I mean, that is synthesized by this
For
unit.
ATP synthesis.
Yes, ATP synthesis. ATP is the currency of
Energy.
uh, energy that is present in the cell, and that currency is actually synthesized by the mitochondria.
Mitochondria. So we can just for our understanding, we can draw a diagram
by which we will try to understand that how it is. So this one is the outer membrane.
Yes, sir.
It is outer, and this one is
Inner membrane crystal.
inner membrane. That is called this inner. These enfoldings are actually crystal. One is crista, so these are multiple grooves are multiple enfoldings. So we
Yes.
call them crystal. So there is another thing that is matrix.
Yes.
Matrix. Matrix is actually it is the fluid part of the mitochondria
Yes, sir.
in which number of chemicals and substances or proteins are present,
Yes, sir.
right?
Yes, sir.
So there is another thing that is DNA. Circular DNA is actually present here.
Yes, sir.
Circular DNA. So we can also categorize circular DNA. Got it? So these dots are what? These are ribosomes because we have studied about it. The ribosomes are also present on these 70s ribosomes. So
Yes, sir.
these are ribosomes. Got it? So
Yes.
let's quickly try to revise all the points related to this. One is double membrane.
Yes, sir.
One is double.
Double membrane, 70s ribosome,
Got
circular
it?
matrix.
Size is what? 1 to 2 micrometer.
Yes, sir.
Yes, double membrane structure. You can add it. So size is what? 1 to 2.
Micrometer.
Uh, yes. Sometimes they change their shape uh, when they are producing ATP. Uh, can you hear me? I think
Yes, sir.
internet. Okay, fine. So during its synthesis, protein synthesis, somehow they change their shape. This is the shape that we have observed. There are many other shapes that we will study specifically in next upcoming chapters. Where they will give you in a diagram where you have to differentiate mitochondria and some other. So mitochondria can also change its shape. So specifically, what's its function? Function. I'm writing here. Function. Function is aerobic respiration. Aerobic
Respiration.
respiration. What type of respiration it is? It is actually definitely the respiration, the breaking down of the uh, substance or the food into smaller particles in order to convert into energy.
Yes.
Definitely we know. For example, if you have glucose, that glucose has to go through this process of respiration, and by the process of this it has to produce energy
Yes, sir.
in the form of ATP. This is respiration, and we will study this topic as well in detail. So its main function is aerobic respiration, produce energy in the form of ATP.
Yes, sir.
Okay. So this is all about uh, these two topics that we have studied. So one is ribosome, what is nucleus, what is ribosome, and how they are differentiated from each other. So then you have another thing that is mitochondria. So
Okay.
if anything you want to ask, so you can.
No, sir.
Okay, fine. So समझ आ रही है बेटा, or is it good for you, or you want to change the medium or anything that you want to suggest, you can. Normally I always teach in this medium just because of the O level student, they should understand this because you have to write in English, you have to
Yes.
interpret, you must so you can communicate easily without
Yes, sir.
with this scenario. Okay,
Yes.
so I think we should close this class today, and we
Sure,
come
sir.
to tomorrow inshallah. थैंक यू वेरी मच। और बस इनको रिवाइज करते जाना है, और कीप
जी
इन
सर।
माइंड, इसको फर्स्ट रिवाइज करते जाएं और ब्रिंग सम। अब मैं वर्कशीट्स भी डिस्कस करूंगा, थोड़ा सा मैं उस तरफ भी आने लगा हूं। जरा मैं आपसे थोड़ा सा हम जरा पेस बनाना चाह रहा था, जरा हम कुछ टॉपिक्स
यस
पढ़
सर।
लें और फिर हम एक कंटिन्यूस। आइडियली तो ये होता है कि 2-3 दिन के बाद आप वर्कशीट जरूर डिस्कस करें, जिसके अंदर
यस
आप कुछ पास्ट पेपर्स के टॉपिक्स जो हैं वो देख लें। आपको भी पता होना चाहिए कि ये क्वेश्चन जो है वो फ्लां पेपर में, बिकॉज यू पीपल हैव लॉट ऑफ स्टफ विद यू, आपको पता होता है ये स्टफ हमारे पास है, तो
यस।
आप देखा करें कि यार ये क्वेश्चन इस टॉपिक से पूछा हुआ है, सो इफ यू डोंट नो अबाउट दैट सो यू कैन आस्क मी। ओके,
श्योर सर।
so that's all for today. See you tomorrow inshallah.
ओके सर, अल्लाह हाफ़िज़।
Goodbye.
अल्लाह हाफ़िज़।
Good afternoon.